Structure of DNA. Before understanding the structure of DNA, we need to understand what is DNA and how do you locate the DNA. So inside a cell, a living cell, there is a nucleus. Inside nucleus, we can see highly contracted, condensed and visible threads of chromatin, which is called chromosome. Now if we zoom in a chromosome, we can see threads of DNA. Now if we zoom in a thread of DNA, we can see a double helical structure, a part of which is known as gene. Now in the DNA, the strand of the DNA is known as sugar phosphate backbone. And inside the DNA, we can see different colored lines which are known as nitrogenous base, in which the red is known as adenine, the yellow one is known as thymine, the purple one is known as cytosine, and the green one is known as guanine. Among them, as we can see, the red one binds with the yellow one. It means the adenine binds with the thymine, and the purple one binds with the green one. It means cytosine binds with the guanine. And all these nitrogenous base binds with each other with the non-covalent bond, which is known as hydrogen bond. Now, if we take a part of DNA, then we can see it is a spirally coiled structure. If we uncoil a DNA, we get two straight lines which are antiparallel to each other. Antiparallel because one of the strand runs from 5' to 3' and another strand runs from 3' to 5'. Now, if we look at the chemical structure of the DNA, we get something like this. In this, the black one is known as phosphate. These blue structures are called 5-carbon sugar, which is also known as pentose sugar. And these colorful structures, they are called nitrogenous base. And the nitrogenous base of one strand binds with the nitrogenous base of the opposite strand with a non-covalent bond, which is called hydrogen bond. It is a breakable bond. And the bond between two nitrogenous base is known as phosphodiester bond, which is a covalent bond and it is a non-breakable bond. Now, in this, the 5-carbon sugar and the phosphate, the chain of 5-carbon sugar and the phosphate forms the backbone of DNA, which means the strand of the DNA. And the phosphate and the 5-carbon sugar and the nitrogenous base, these three combines to form nucleotide. And the nitrogenous base and the pentose sugar, these two combines to form nucleoside. Now let's look at these structures in detail. Here in one number you can see there is a phosphate, there is a pentose sugar and there is base. So they are nucleotide. In second figure you can see there is a pentose sugar and base. So they form nucleoside. In third figure you can see there is only nitrogenous base, single ring structure and double ring structure. So they are base. And in fourth figure you can see pentose sugar and phosphate, a chain of pentose sugar and phosphate, which forms the backbone of DNA, that is chain of DNA or a single strand of DNA. Now let's come to the bonds. We can see there are two nucleotides and they bind together with a bond called phosphodiester bond, which is a covalent bond and it is a non-breakable bond. Now let's come to the nitrogenous base. So what is single ring nitrogenous base and what is double ring nitrogenous base? So single ring nitrogenous base are those base that contains one hexagonal ring, whereas double ring nitrogenous base are those base that contains two ring. Single ring nitrogenous base are also called pyrimidines because they contain pyrimidine nucleus, and double ring nitrogenous base are also called purines as they contain purine nucleus. Pyrimidines are thymine and cytosine, whereas purines are adenine and guanine. It means thymine and cytosine are denoted by single ring, whereas adenine and guanine, they are denoted by double ring. And one thing to know is, in a DNA, purine of one chain always binds with the pyrimidine of other chain, but adenine binds with the thymine with double bond and guanine binds with the cytosine with triple bond, due to which the adenine and thymine bond is more easily breakable than that of the guanine and cytosine bond. So in a DNA, number of purine is equal to number of pyrimidine. Coming up to base pair. What is base pair? When a nitrogenous base of one chain binds with the nitrogenous base of opposite chains, it forms a base pair. In short, when a purine of one chain binds with the pyrimidine of opposite chains, it is called a base pair. Now let's see why the DNA strands always run from 5' to 3'. So in a pentose sugar there are 5 carbons, and if we count, then we can see one phosphate is always attached to the 5th carbon, and one phosphate is always attached to the third carbon. And if we elongate the chain on both the side by adding nucleotide chain, then we can get two free ends, one with 5 prime and other with 3 prime. So the chain of DNA always runs from 5 prime to 3 prime. As we can see in this figure, if we consider this as the midline, then we can see one strand of DNA is running from 5 prime to 3 prime and other strand of DNA is also running from 5 prime to 3 prime, but in opposite direction. So the chains of DNA are antiparallel to each other. Now some basic things to know is the distance between two strands of DNA is 20 angstrom, and the distance between one complete 360-degree round of DNA is 34 angstrom, which means the distance between two hydrogen bond is 3.4 angstrom, which means that in a 360-degree round of DNA there are 10 hydrogen bonds. 3.4 multiplied by 10 is 34 angstrom. So in a complete round of DNA there are 10 hydrogen bonds. Now one thing to know is between two hydrogen bond there is an interaction called pi-pi interaction, just to know. Now the grooves. In a DNA, due to coiling of the structures, various grooves are formed. Some of the grooves are larger, some of the grooves are smaller. The larger grooves are also called major, which is 22 angstrom in length, and the smaller grooves are called minor, which is 12 angstrom in length. And the function of these grooves are to bind the protein inside them. Now let's come to our last topic, codons. So 3 nitrogenous base together constitutes as codon, and the function of codon is to code for a single specific amino acid. Let's take an example of some bases. These are some bases in our DNA and RNA. Among them, A, U, G means adenine, uracil and guanine. They code for an amino acid called methionine, and G, G, G they code for an amino acid called glycine. Now we can see that there are total of 64 codons in a DNA sequence. Among them, 61 are called sense codons and 3 are called nonsense codon. Sense codon because they code for amino acid. Nonsense codon because they do not code for any amino acid and they terminate the sequence. Now there are total of 20 amino acids in our body. So if we do the numbering, we can know that for a single specific amino acid there is more than one codon, but one codon only codes for one specific amino acid. So one codon codes for a single amino acid, whereas one amino acid is coded by more than one codon. To understand this, let's take an example. Let's consider these three as codons, which codes for a single amino acid. So here we can say that all these three codons codes for a single amino acid. Individually, if we see, we can understand that this specific codon codes for this specific amino acid, but this specific amino acid itself is being coded by 3 different codons. So that's all about the basic structure of the DNA. Thank you so much for watching.